Advances in Precision Therapeutics. Please welcome our session chairs, Dr. Tan Meng Ho, Associate Professor, School of Biological Sciences, Nanyang Technological University, Singapore, Dr. Pratibha Madapura, Senior Lecturer, Institute of Dentistry, Queen Mary, University of London, UK.
All right, let's get started. So welcome to the session on Advances in Precision Therapeutics. So the first speaker is Dr. Rajkumar Banerjee, who is a Chief Scientist and Professor at CSIR-Indian Institute of Chemical Technology, Hyderabad. His research focuses on lipid-based delivery systems for non-viral gene and drug delivery, anti-cancer nano-therapeutics, and nucleic acid delivery for vaccines and genetic disorders. He has published over 100 papers, holds 19 patents, and has guided 25 PhD students. Dr. Banerjee is a recipient of the CSIR Young Scientist Awards, the IUSSTF Visiting Scientist Fellowship, and the CDRI Award for Excellence in Drug Discovery. Today he will talk on glucocorticoid receptors, a potent target for drug sensitization, immuno-modulation in nano-therapeutic. Please go ahead.
Thank you. Thank you for the kind introduction, and thank you everybody here for your kind invitation, impeccable sense of hospitality, especially food, and I don't know, this inventive desserts with coconut milk. So old thing, but yet, anyway, I will basically talk about glucocorticoid receptor. It's also a very old receptor to talk about. This receptor and a molecule called dexamethasone, which is like 50, 60 years old, and people never thought possibly that it can be used so effectively to do the immuno-modulation. So this word immuno-modulation and drug sensitization, frankly speaking, this is what I'm doing for last almost 20 years, and all my PhD students, I don't want to name them specifically right now, they have one chapters at least they spent for their PhD research on glucocorticoid receptor. So, but glucocorticoid receptor actually is a cytoplasmic receptor. But if you see here, we started our career with targeting cancer by targeting the cell surface receptors like sigma receptor, folate receptor, etc. And that's what everybody does. But then we thought that let's try with some estrogen receptor. That was long back, 2005. But of course, estrogen receptor and glucocorticoid receptor is another cytoplasmic receptor. But there is a difference between the use of estrogen receptor and glucocorticoid receptor. Estrogen receptor would be expressing only in specific cell types, but glucocorticoid receptor is not. Anyway, when we switched over to glucocorticoid receptor targeting, God willing, we had a lot of papers. So I just wanted to tell you, it's a very simple system. All you have to do is to target glucocorticoid receptor by associating this molecule called dexamethasone in the lipid bilayer. So, but glucocorticoid receptor, as you know, you may be knowing, it's also called nuclear hormone receptor. The hormone should come and sit into this receptor in the cytoplasm, go inside the nucleus, and that's the reason they are called nuclear hormone receptor. But there is a challenge. It actually expresses in almost all cells. And so, cancer-specific targeting is hypothetically not achievable. Compounding this factor, if you see this red boxed area, prolonged use of glucocorticoids, mostly the glucocorticoids used as palliative medicine, that leads to resistance against itself and treatment drugs through peak glycoprotein upregulation. So that means if we use it for a prolonged time, there will be a lot of peak glycoprotein coming up, and it will make the glucocorticoid receptor move away from the cells. So, it makes the cells so much resistive against many drugs. Corticosteroids lead to chemoresistance is a well-known fact. With this background, it is very clear that it's a big challenge to target glucocorticoid receptors. But then, our idea is to target cancer, and that too through glucocorticoid receptor. Let's look into these blue ones: immune function, growth development, and gluconeogenesis. Clearly, immune function, growth, and development, all these are like a beaten down words that we often see for cancer treatment. Clearly, this actually fascinated us. But then, what about gluconeogenesis, the alternate source of energy for cells? And glucocorticoid receptor precisely does that. But how is it important? So, if you understand this schematic diagram, students here would understand very well why we actually chose glucocorticoid receptor as a potent target. What actually happens, the glucose converts into pyruvic acid, as you know, through glycolysis, right? And that, the pyruvic acid has two ways that it can go to, either form lactic acid, or it can goes into this OXPHOS, the mitochondrial oxidative phosphorylation, to form 32 ATP. Keep it in mind, cancer cells would really mind having 32 ATP in one shot of experiment, because that excess ATP will actually stop the glycolysis. If that happens, then actually cancer cells cannot survive. So they find that they are very selective. The use of OXPHOS for cancer cells is very selective. And that actually happens only in differentiating cells. Differentiating cells prefer OXPHOS, but the proliferating cells, like cancer cells mostly, they prefer glycolysis. Now, this pyruvic acid and lactic acid, this lactic acid, more the lactic acid in the cell will become acidosis, will have the acidosis property, and that's bad for the cell, and so it has to throw away the lactic acid from the cell. As a result, this lactic acid will be used for metastasis and invasion later on, but while pushing the lactic acid out of the cell will grow the P glycoprotein, the efflux pumps, and etc. And that is what actually tries to induce the drug resistance. Then what we can do, and what is this gluconeogenesis? Gluconeogenesis, the starting point is pyruvic acid and lactic acid. So if you can force the cancer cell into gluconeogenesis, it will go back to glucose. But just please look into this. It shows that minus delta G glycolysis is always greater than minus delta G gluconeogenesis. That means gluconeogenesis and glycolysis both can happen because they have minus value, right? It's basically a free energy that is released out of the body. But because delta G glycolysis is more, that means if I forcefully do the gluconeogenesis inside the cell, actually I am expending the energy out of the cell. I'm making it a little more sensitive, and that's precisely what we are doing. And how you can do that? So, as you can understand, the last line, if you can't see it from the back, glycolysis is preferable energy metabolism in cancer cells. So, what magic we did? Very simple. We took cationic lipids, and that's what basically people do. They take cholesterol to fortify the liposomes that is to be formed. The bilayer will have these two. And if you want to make it target to glucocorticoid receptor, all you have to do is to just take this dexamethasone and associate along with this. See the structure, both are steroid in structure, and it can be easily accommodated. And no wonder it started showing, this simple formulation started showing some wonder, and we got a US patent out of it. Who gives a US patent to a molecule just making a formulation? Now, because of its use. What we find is very simple. I'm not ashamed to show a very old data of mine, which is 2009 molecular therapy paper, where we showed that the targeted formulation which contains dexamethasone actually can access inside the nucleus of the cancer cells, but they have no access inside the non-cancer cells. But if you take just the formulation which does not have dexamethasone, cannot go inside the nucleus. No way. But then, what happens if it goes inside the nucleus? If it goes inside the nucleus, what a pure dexamethasone does, the formulation also does the same thing, but only in cancer cells. but not in non-cancer cell. That means you can have a easy and selective access to preferred sites of cancer genome, because this CYP3A5, just as a model we used it, this CYP3A5 is basically a metabolizing gene, but it's basically a signature gene to show that whether our formulation has access to the nucleus, and that to the glucocorticoid response element. If this is happening, and we find that if you put dexamethasone, you see there is a little bit upregulation compared to untreated, but in if you use the formulation, there's a huge, it's like 1.5 to 10 folds more of upregulation. But if you take it in a non-cancer cell, untreated doesn't have anything. If you put dexamethasone, the same value we got it for dex formulation as well. So that means the cancer cell and normal cell, they can differentiate in recognizing the formulation itself. If this is in mind, let's try to see, is nuclear localization in GR targeting strategy useful for targeting and treating cancer stem cells? Now the question is, what does a cancer stem cell show? Just there are many properties: inhibition of self-differentiation, highly drug resistance, and of course they are regulated by dysregulated developmental pathways. What actually happens as just a cell biologist, what they look into, they see, as far as drug resistance is concerned, the P glycoprotein will be upregulated, MDR genes will be upregulated, and this ATP binding cassette transporter in stem cells, or BBB, this ABCG2, whatever, it actually is upregulated. Inhibition of differentiation, one factor, it upregulated. All we have to do is, using this formulation, can we reverse that? That's what we wanted to show. So there are many work on it. I'm not going into the details into that. I just wanted to show you if we can utilize this formulation to repurpose a molecule like 5-fluorouracil is a first generation drug used for a colorectal cancer patient's first generation formulation, and it is nowadays not even used because it is of no use. The cancer cells have become resistant to 5-fluorouracil. They are basically using these multi-drug regimens. Folinic acid, 5-fluorouracil, and oxaliplatin, it's called Folfox. Or if you use irinotecan, it's called Folfiri. So this is what actually colorectal cancer patients are being treated. Now if you use just simple 5-fluorouracil, not even the other ones, just take 5-fluorouracil in our formulations. So I just put lipids, there are many lipids in it, but dexamethasone extra is there, and the control does not have dexamethasone. And you see the first observation that it actually goes inside the nucleus of the colon cancer cells, okay? But, and this is the human colon cancer cell also, we have seen that it goes inside the nucleus, but the same formulation does not go, if you use the control formulation, it does not go inside the cancer cells, whether it's of human or non-human in nature. But then this formulation, targeted formulation, does not have any access inside the cancer of the non-cancer cell. Clearly, the formulation that we made is showing the first observation whether it goes inside the nucleus or not. Yes, it is good. 3 examples. One example is this, where a first generation anti-colorectal cancer drug 5-fluorouracil can be repurposed using this GR targeted formulation. So what we did was, so the GRE regulation gene CYP3A5 is, as usual, it is going up, okay, but it is not happening inside a non-cancer cell. So it is now actually falling into that 2 points. One is nuclear localization and the CYP3A5 upregulation is happening. Then we developed this colon cancer model in situ. Our chemist students, they learn the surgery and they do it. And then we treat our formulations, and you see the D1X5FU, that is the targeted formulation, you have a huge E-cadherin upregulation, clearly showing there is an EMT reversal, and there is a downregulation of hemantin. That means we are actually reversing the EMT just using this formulation. And you have a downregulation of MDR1. See the formulations, these are being used in the clinic. Folfiri and Folfox, they are 3 drug system. And you see that there is no change in the MDR1 pattern, but in our formulation it's actually going down. So we just injected every alternate day 5 times, this only 5 times, and then we wanted to see how long our targeted formulation can keep the mouse, longer, whether they are having their longer survivability or not, and that's what we are finding. But I just wanted to show you this data. Please have a little closer look. When we stopped our injection here, and then we sacrificed some representative mice, we find that this is the healthy cecum which does not have any tumor, and you see that the D1X5FU actually has a very little amount of bulge there. Very little amount of bulge. I don't know whether you can see it, but I know it that there is one. But in case of even the fall fox or fall fury, there is a huge bulge. And after that, we had to stop the injection just to see how long it stays. So clearly, the first generation drug can be repurposed in this platform formulation. So you can actually regenerate the formulation. All the existing, I'm not talking about, we have actually used 25 different drugs, more than 25 drugs, or more maybe, I don't remember now. But this is one just example to show that an old drug that is now not been used, but is still in clinics as a multi-combination formulation. You just put it into this platform and you can repurpose it again. And it's much better than the existing combination. But the question is, we as a chemist, we always look to do some chemistry, It's just not that associating lipids and steroids and dexamethasone sort of thing in a associative formulation like liposome. So we thought that instead of liposome, can we conjugate the glucocorticoid ligands with cationic lipid? So are there instances? Yes, for last 25 years, we did a multiple of molecules. So this is estrogen, this is progesterone, estrogen again, dexamethasone, lithocholic acid. This is the work with, you remember, estrogen, lithocholic acid. This is hydrocortisone, then we have folic acid, haloperidol, and these oxindole molecules. We tried a lot, and you see the common thing is these red ones, a cationic lipid there, short chains, not very big. And you just go into this literature and you'll find they are actually making these molecules repurposed. Progesterone, you cannot want it to be a drug, right? Progesterone is an endogenous hormone. You just convert it into a drug. Like that, a simple cationic lipid conjugation does that. Here, I actually focus only on dexamethasone or glucocorticoid receptor targeting. So here is dexamethasone based as well as hydrocortisone based. This is a different story to tell later, but I will just give you this example. Hydrocortisone. So what we did, simple, we took the hydrocortisone, do a simple conjugation with a cationic lipid. Okay, made a series of it, 8, 10, 12, 14, and 16, and we find that C16 molecule clearly has a anti-tumor effect. Okay, this is a huge size, I'm sorry, but I just wanted to tell, it has on its own anti-cancer property, but what else are we seeing? If you use the molecule at a concentration like 2.5 and 5 micromolar, it does not have any toxicity. But if you use, and the doxorubicin has some IC50 around 1 micromolar here, right? But you put these two molecules into it, you see the IC50 values. I don't know, it's much lesser than maybe 10 or 20 nanomolar. It's like a very huge difference, clearly showing that even doxorubicin can also be repurposed if it is being used. But how can you use it if you want to co-deliver it? We find that high C16, if you do a simple ethanol injection process, it's a very simple process. Anybody who can visit my lab, I will show you how to make it. It's very simple. This ethanol injection process makes a self-aggregating molecule with an average diameter of around 180 and a positive charge of 32.9. But is the molecule is localizing inside the nucleus? It is. The CD15 and CD26 are two colon cancer cells, but it is not inside the CHOC. Even the molecule is showing the nuclear localization, the molecular hydrocortisone. The hydrocortisone is a molecule which is an endogenous ligand, right? And you convert it into a hydrocortisone cationic lipid conjugate. So, because of its positive charge and it's an aggregate in nature, certainly you can deliver p53, a anti-cancer gene, and you see that the hyocysteine with p53 has the better survivability. It's around 54 days. But because it's an aggregate, it can deliver a gene as well as a drug in combination. What we are seeing is a clear increase in the survivability, and the size is almost, this is a healthy cecum, and this is our treatment. Now I go to this part, how can I modulate immune function by GR transactivation? So here, the immature myeloid cells, you may know that the STAT3 through NF-kappa B and PI3 can STAT3, these are the pathways which actually activates into these MDSCs, the myeloid-derived suppressor cells. And it has its function, inhibition of NK cells, B cells, etc., right? And converting M1 to M2 macrophages, Treg upregulation, so, and the T cell suppression and all that. So what we did was, as Jayanto in the last talk clearly showed, I'm not showing here, you isolate the dendritic, the bone marrow cells, and then you treat with our formulation, engineer them and put it back to the mouse. This is the data after this is basically a agglomeration of, after a lot of deliberations and experiments, we've come up to this. So what we did was, first we induced CD26, the colon cancer cells, and then we put our engineered cells, and then we treated with our liposomal formulation. And we see that the tumor never actually went up. I did not show it for a longer time, but it has actually went up like this. So it's only like 7 or 8 injections. And clearly, there is a downregulation of PD-L1 in the tumor. That even niclosamide, oh, here, I forgot to tell you, this formulation actually is with niclosamide molecule. Where did I write out? So N means niclosamide. Okay, so niclosamide has its own, it's a STAT3 inhibitor, and it has its own PD-L1 downregulation, but does not have any effect on tumor. But you put it into the formulation, you have a huge downregulation. And here you see that it is actually acting through GR, as it's shown by I-kappa B upregulation, NF-kappa B, and phospho-NF-kappa B downregulation. But this is the one I thought that I should talk to you a little more. You see that the M1 population in the tumor is going up. So, and this is the M2 population, CD206, has gone down. So a ratio of M1 by M2 within the tumor is actually tremendously high. Clearly, it's anti-proliferative in nature. Now, if you see, the CD4 population is actually going up, as well as the cytotoxic CD8+. And as a proof, within the tumor, we find that the arginase actually is going down. And this is actually, arginase actually works on the arginine, which is the food for T cell proliferation. So clearly, we are inducing tumor. And this is the data where Treg actually is going down compared to the Treg in an untreated or in even niclosamide treatment or in a non-GR targeted niclosamide formulation. So it's a very clear downregulation, and this is actually compounded by this Th1 upregulation and Th2 downregulation with a overall Th1 and Th2 upregulation. These are all data in the tumor that we isolated. So clearly, the formulation has its effect against the cancer cell as well as the MDSCs within the tumor. But here we actually re-engineered these MDSCs, and that actually did the trick. But then I'll just end with my data with this, some clinical samples. And this is done in collaboration with Mayo Clinic USA. What they did was, so this is my third example and the last example, and this is the last data. Don't worry, I won't take much time. I'm almost there anyway. So what they did was, they took 2 patients, and these 2 patients, they are actually CLL patients, chronic lymphocytic leukemia patients, and they are bortezomib resistant. Let's see if this bortezomib we put into our formulation, how they work. So what we did was, we took the normal healthy donor PBMCs, and we also took the CLL patient blood cells. The idea is, check your formulation and find the IC50 value against this PBMC and these cells. If you have a ratio, IC50 fold difference between PBMC by patient cells is higher than 2, my collaborator in Mayo Clinic, who is basically a hemato-oncologist, he told that if it is more than 2, your formulation actually can be used for clinical trials, at least in small cohort of patients. What we are seeing here, take the formulation D1X. Now this first patient, you have a ratio of 1:0. That means this patient is highly sensitive against the formulation, which is just a platform, it does not have the drugs. But this patient is not working only with the dexamethasone formulation. But you start putting this bortezomib or Velcade, you see the ratio 24 and 12.6. You use, this is our endogenous molecule, the structure I showed you, ESC8, but I did not talk much about it. You see the now 196 of 108. You use a molecule Nutlin-3. Nutlin-3 is basically, it acts against this MDM2 and it inhibits the MDM2-P53 interaction. This actually is a failed drug in phase 3. And we find that even using this Nutlin-3 in our formulation, it actually can resensitize the patient samples, the CLL patient samples. And so, can we say that the GR targeting concept can be used as a platform for personalized medicine? Because this data clearly shows that some patients are quite more sensitive to certain formulations and some are less. So, depending on what drug would you use, you just take the patient sample and do it. This kind of study. So to end with, first, yes, thank you. Thank you, I came into the conclusion part. So the glucocorticoid receptor targeted platform formulation is a for multifaceted anti-cancer application. First, drug repurposing. You use this formulation to repurpose a first generation drug. Second, using a molecule like niclosamide is also a very cheap drug. You can see it in your local pharmacy. Put it into our nano formulation, and we did the same DSCs and stem cell reengineering. I did not talk about stem cell reengineering, it's a work of Pritham, he's sitting there, but maybe next time. And of course, you can use it against the blood cancer as possibly a personalized medicine platform. So to acknowledge, as I told, all these students, they actually have at least one chapter in their thesis, they work for glucocorticoid receptor. But frankly, Amarnath and Pranav is sitting here. He is now professor in BITS. He was my first postdoc. So Amarnath and Pranav actually, they principally showed that nuclear localization, signaling and all that. And sensitization is shown by Sujan against the cancer stem cells. Bhomira and Kattayani, they actually, they worked on this hydrocortisone cationic lipid conjugate. Namita showed this 5-fluorouracil part. Tithi showed MDSC work. And Pritham cancer stem cell, I did not talk much about, but she also actually helped Tithi on that MDSC work. I'm very much thankful to all my granting agencies. I don't have very flowery pictures for each agencies, because I have to accommodate them here. And Professor Deb Mukhopadhyay from Mayo Clinic, and of course Pranab Narayan, who is actually in many of my later projects. Anyway, nobody actually talks about the students who are actually helping us during this whole symposium, right? We always talk about the Srujan and Srujan et al., the colleagues. So here is a small couplet for the students. So dexamethasone is basically one big scientist in India once told me that dexamethasone and glucocorticoid receptor is a beaten down thing, it does not have any future. But then there is a darkness around me some time back, but I see that there is a light everywhere. Thank you so much.